Amino acids has two functional groups, amine and carboxylic acid. In the case of natural amino acids, both the amine and the carboxylic acid is bonded to a common carbon. That carbon is termed as the alpha carbon. There are 20 natural amino acids, so there are 20 different R groups that's bonded to the alpha carbon. Physical properties of amino acids. Amino acids exist as Zwitter ion. Zwitter ion means dipolar ionic species, meaning to say they have one side that is actually positively charged, another side that's negatively charged. Hence, they're actually ionic in nature. Hence, the Zwitter ion are all closely packed together in a giant ionic lattice structure. Hence, it has high melting point and boiling point. In the presence of water, it's able to establish ion-dipole attraction forces, so amino acids are highly soluble in water. Finally, amino acids is able to be separated by electrolysis based on the pH of the medium. So let us take a look at some example. When amino acids exist in a high pH medium, then the NH3+, which is a proton donor, behave as an acid and neutralizes with the OH- to become NH2. So at high pH, the amino acids will be negatively charged. Let's take a look at what happens when it's a low pH. At low pH, the carboxylic ion will behave as a base to neutralize the H+. Therefore, as a process, it will form COH. Chemical properties. For the chemical properties of amino acids, we'll first cover the acid and then the base behavior. As earlier illustrated, the NH3+ is the acid group in amino acids, while the carboxylate is the anion group. Let's see what happened initially in a sample of your amino acids when the pH is low. So at this low pH, in the conical flask, the structure of the amino acids will be as such, which means it is a dibasic acid because the carboxylic acid and the NH3+ could actually neutralize the OH-. And as you add your alkali, COH being the stronger acid group relative to NH3+, will neutralize the OH-. Hence, at this pH, the COH will be neutralized to form COO-. This is also the point where amino acids becomes a Zwitter ion. And at the pH where the amino acids becomes a Zwitter ion, which means the molecule does not have any charge, that point is termed as the isoelectric point. So specifically for this example, the pH of the isoelectric point is actually 6.02. When alkali continue to be added to the amino acids, you'll find that now the NH3+ will be neutralized to becomes NH2. So at this pH, this is when you get this structure. Finally, it's important to note that there are two buffer regions. The first buffer solution comprises of these two forms of amino acids, where in this case, this is the acid and this is the base. At this specific pH of 2.3 is actually pH equal to pKa1. So Ka1 is the dissociation constants of your carboxylic acid. In the second buffer region, it comprises of the Zwitter ion and this form of amino acids. So in this case, this would actually be the base, while the NH3+ is the acid. And at maximum buffer capacity, the pH will be 9.7. That is when pKa2 is equal to 9.7. So your Ka2 is referring to the NH3+. It's important to note that your Ka1 is the first dissociation of the amino acids, specifically for the group COOH, while Ka2 is the acid-base dissociation constant for NH3+, and in this case, it's a second dissociation of this amino acid. And finally, the pH of the isoelectric point, which is your 6.02, is in actual fact equal to half the value of your pKa1 plus your pKa2. A good question then to ponder about is that is an amino acids in the form of Zwitter ion, is it amphoteric? The answer is yes, it's amphoteric because it can neutralize H+, it can also neutralize OH- as observed here. And the next question to ask is that amino acid as a Zwitter ion, is it a buffer solution? The answer is no, because a buffer solution is defined as a solution that can best resist pH change. Certainly in the form of a Zwitter ion, when a small amount of your OH- is being added or H+ is being added, there's actually a big change in terms of a pH. Hence, amino acids in the Zwitter ion form is not a buffer solution. However, These two forms of amino acids, when exist in equal amount, can form the maximum buffer cap, and the pH of the maximum buffer cap solution will be 2.3. All the amino acids in these two forms, when exist in equal amount, can form a maximum buffer solution, where the pH of the maximum buffer solution will be 9.7. Before we cover the last chemical properties of your amino acids, let's take a look at electrolysis of an amino acid. Given that the pH of its isoelectric point is 6.06. What that means is that at this point, the amino acid does not have any charge. It exists purely as a Zwitter ion. So in this case, when actually subjected to a positively and a negatively charged plate, the amino acids stays at where they are. It does not move. However, if you put them in a solution where the pH is more than the pH of the isoelectric point, the NH3+ group in the amino acids will be neutralized to form NH2. And therefore, because of this, the form of the amino acids will be negatively in charge. Therefore, it is an anion. So during electrolysis, the anion will then move towards the positively charged plate, which is the anode. Finally, if the pH is less than the pH of your isoelectric point, which is 6.06, then your COO minus will now accept the H plus to become COOH. After it becomes COOH, the form of the amino acids that exist will be positively charged. So in this case, it will be a cation. So the cation will be attracted towards the negatively charged plate, which is the cathode. The final reactions that we've already learned is that two amino acids can be catalyzed by the enzyme. And in biochemical reactions, the oxygen and two of the hydrogen molecules will combine, forming your water, and the process forming an amide bond, or otherwise what is termed as a peptide bond for biochemical reactions.